Hello everyone, welcome to my channel. I am Shweta and today we are going to see pyruvate oxidation. For better understanding, please watch my video on glycolysis before watching this video. We all know that glucose gives pyruvate in the process of glycolysis. This is partial oxidation of glucose. Now, what is the fate of this pyruvate? If the molecular oxygen is present, we have seen that in cell aerobic respiration takes place and this pyruvate undergoes further oxidation. The site of the pyruvate oxidation is mitochondrial matrix. Site is mitochondrial matrix. Now let us see the reaction. Pyruvate plus coenzyme A gives acetyl coenzyme A. In this process, carboxylic group of pyruvate is released as CO2 and NAD plus acts as the oxidizing agent which oxidizes pyruvate and itself is reduced to NADH. So the 2 carbon of the pyruvate gives the acetyl part of acetyl coenzyme A, and this reaction is catalyzed by enzyme pyruvate dehydrogenase complex. So from this we can say that here carbon dioxide is released and the oxidation takes place. So it is a oxidative decarboxylation process. Second, it is an irreversible process. It is highly exergonic process. That means in this process large amount of energy is released. Now let us see about pyruvate dehydrogenase complex enzyme which catalyzes this reaction. It is an assembly of 3 individual enzymes: pyruvate dehydrogenase denoted as E1, dihydrolipoyl transacetylase denoted as E2, and dihydrolipoyl dehydrogenase denoted as E3. These involve certain coenzymes like thiamine pyrophosphate, lipoic acid, coenzyme A, FAD and NAD+. Now let us see where it is located. This is mitochondria. Outer mitochondrial membrane, inner mitochondrial membrane, intermembrane space, mitochondrial matrix. this is cytosol. So the enzyme is located on inner mitochondrial membrane, E1, E2, E3. Through glycolysis, pyruvate is produced in cytosol and then it enters the mitochondrial matrix, and here the entire reaction takes place. Let us see the entire reaction in detail. Pyruvate dehydrogenase complex is an assembly of three individual enzymes: pyruvate dehydrogenase denoted as E1, dihydrolipoyl transacetylase denoted as E2, and dihydrolipoyl dehydrogenase denoted as E3. Coenzymes are thiamine pyrophosphate, lipoic acid, coenzyme A, FAD and NAD+. Pyruvate dehydrogenase along with TPP catalyzes the decarboxylation of pyruvate, CO2 is released and hydroxyethyl thiamine pyrophosphate is formed. Dihydrolipoyl transacetylase then converts hydroxyethyl thiamine pyrophosphate to acetyl lipoamide. This then transfers the acetyl group to coenzyme A and forms acetyl coenzyme A. Lipoamide is obtained in reduced form. This reducing equivalent is then transferred to FAD and forms FADH2, that is catalyzed by dihydrolipoyl dehydrogenase, and lipoamide is obtained in oxidized form. FADH2 then transfers its reducing equivalent to NAD+ and it forms NADH plus H+. This then by electron transport chain through oxidative phosphorylation gives 2.5 ATP. Now this acetyl coenzyme A enters into Krebs cycle. So this reaction is known as link between glycolysis and Krebs cycle. Now what are the functions of these individual enzymes? Here we see that CO2 is released from pyruvate. That means decarboxylation of pyruvate is taking place. Thus pyruvate dehydrogenase catalyzes the decarboxylation of pyruvate. In this step, acetyl group is transferred to coenzyme A to form acetyl coenzyme A. Thus we can say the dihydrolipoyl transacetylase catalyzes the transfer of acetyl group to coenzyme A. Now here we can see that the reducing equivalent of lipoamide is transferred to FAD and it is self-oxidized. Thus dihydrolipoyl dehydrogenase catalyzes the reoxidation of lipoamide. Thus we can say that pyruvate dehydrogenase catalyzes the decarboxylation of pyruvate. Dihydrolipoyl transacetylase catalyzes the transfer of acetyl group to coenzyme A, and dihydrolipoyl dehydrogenase catalyzes the reoxidation of lipoamide or lipoic acid. This is all for pyruvate oxidation. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.